It's a pleasure to introduce Dan Lippman as the inaugural Jack Strominger Lecture. But before I do so, a few words about Jack. I've known Jack for the better part of 30 years, I think, and I know him both as a mentor, a teacher, and above all, a family friend. It's a very special relationship where Jack has received me in his home on multiple occasions. We've had occasion to discuss family affairs that should be left private. But in all of this, he's been a stalwart and a strong supporter throughout my career, and not just mine, but those of many of his trainees. I don't think we need to spend a lot of time on Jack's scientific accomplishments, starting out at the NIH and then through WashU, St. Louis, Madison, Wisconsin, ending up at Harvard University, where his discoveries are really textbook lore. We know about nucleotide sugars and their involvement in bacterial cell wall synthesis owing to Jack's work. We know about the mechanism of action of penicillin through Jack's work. He started a wonderful collaboration with crystallographers to elucidate the structure of the histocompatibility antigens, and these have now become the subject of tote bags, coffee mugs, and T-shirts of immunology graduate programs. And these discoveries have all been richly rewarded by recognition, by learned academies, prizes, amongst which the Lasker Award, the Japan Prize, and many others. Jack is now 91 years old, and at his 90th birthday, we had a symposium that celebrated his accomplishments. Thank you for the musical accompaniment. And on the occasion of his 90th birthday, it was decided that it would be a good idea to honor Jack in a more lasting manner, and that's what led to the establishment of the Jack Strominger Lecture. We have generous support from the Department of Stem Cell Biology, Stem Cell and Regenerative Biology, the Evergrande Foundation at the medical school with generous contributions from their leaders, Arlene Sharp, Vijay Kutru. Gerald Chan is here, who has been likewise a generous supporter, and many of Jack's friends have pitched in to make this hopefully a lasting event. Now, what I'd like to show is, if I only knew how to do that, this is a picture that is one of my favorites. This is Jack and Ann about 20 years ago at their farm in East Alstead. Those of you who visited the farm and have been received by the family will recognize the warmth and generosity that accompanied these visits. This is another image, a very typical one of Jack mentoring a student. In this case, this is myself. Some 20 years earlier, discussing a paper which obviously had some critical nuts to be cracked. But it's a pretty typical image, I should say, and those of us who've worked with Jack on manuscripts will recognize instantly the frown on his face, the skeptical look, but in the end, these things end up rather well. So enough about Jack. The first lecture we chose was somebody who had roots in developmental biology, immunology, and more recently in aspects of public health, such as they emerged from the study of the microbiota. The speaker, of course, is none other than Dan Lippman. He worked at UCSF as a graduate student. As an undergrad, he even published some papers with Mark Kirschner on microtubule-related stuff. I didn't know this until I searched on PubMed what he had done. As a postdoc with Richard Axel, he did seminal work on changing the nomenclature of all these complicated immunological surface proteins. T8 became CD8. And then later, independently at the Skirball Institute, he performed that as a true art form, uncovering locus control regions, studying regulation of expression of CD4, CD8 surface markers, making key contributions to understanding of T-cell development. That is a trait he shares with Jack, I should say, this continuous ability to reinvent himself. Not only did he work on gene expression of these surface proteins, he also studied T-cell development, and then later on, the relationship of HIV with chemokine receptors, HIV with CD4, the discovery of DC sign as an important entry receptor for HIV, and then of late, of course, ROR gamma T, transcription factors that control the development of Th17 cells, and this is truly an area that Dan has pioneered. This then led to his interest, which continues to this very day, of the microbiota and its interactions with T-cell compartments, and that's what he's going to talk about. So without further ado, once again, I thank the supporters of this lecture for making all of this possible. I thank Gerald Chan for hosting us at some fine dining establishment in Cambridge. And I thank, above all, Dan Littman for the willingness to serve as the first Strominger lecturer. Dan.
Thank you so much, Hida. It's really very humbling to be here as the first Jack Strominger lecturer, because Jack has been a hero of mine now for some 40 years. There is a pointer here, right? Yeah. And Jack probably doesn't know it, but I am a scientific great-grandchild of his, because I did my PhD with Ben Schwartz and Susan Cullen, actually in St. Louis at Washington University, and they were mentees of Stan Nathanson, who was a postdoc with Jack in the 1960s. So we have a connection, and that connection actually continued for a number of years without probably Jack even knowing it, because for my graduate work, I took on a project that was really really quixotic and sort of foolhardy. But it turned out, for reasons probably different, Vic Engelhardt in Jack's lab was doing a similar kind of project. And that was to try to reconstitute MHC molecules into lipid membranes, into liposomes. I was doing it to try to understand how the T-cell receptor recognizes nominal antigen and MHC. At the time, a really central question, and it was a completely flawed experiment because it would have never worked. But we did publish papers, both of us. Vic before me, and then a little bit later, I published this paper as part of most of my graduate work. Now, the reason that my experiment was so flawed became very obvious in a pictorial way a few years later, when Pam Bjorkman, a student of Jack's, with Don Wiley, solved the structure of the MHC class I molecule. So the antigen that I was looking for in a liposome, either the same or a different liposome, turned out to be a peptide embedded in the MHC molecule, explaining very clearly why my experiment was kind of stupid. But I showed this because this was so revealing to so many of us in the field of immunology. We knew about a lot of very beautiful work on MHC restriction and the biology of recognition of specific antigen by T lymphocytes. But at that time, we had really no idea what the T-cell receptor that had been solved a couple of years earlier, what that was actually recognizing. And all of a sudden, with this one picture, everything became so obvious. To me, this is still the most revealing crystal structure that I've ever seen in terms of explaining biology. And it's really to Jack's credit that his many years of work led to that. My interactions with Jack or his lab continued because I went to do a postdoc with Richard Axel, and I wrote a fellowship proposal from Jankov and Childs to use gene transfer to clone MHC class I molecules. And about 3 months after I started my postdoc in Richard's lab, this paper came out. Thanks, Ida. I realized that I had to change course. So I changed course to work on T-cell surface molecules, and I was fortunate that I could clone CD4 and CD8 by gene transfer as a postdoc. And then in my own lab at UCSF, I started working on what these molecules do. And here again, I had an interception with Jack, because Carolyn Doyle in his laboratory was working on the interaction of CD4 with MHC class II, and my first MD PhD student, Ann Norman, in my lab was working on CD8 interaction with MHC class I, and these made very nice bookends to understanding how co-receptors complement the TCR specificity in the interaction with MHC molecules. So it was then that I decided I really had to get away from Jack. So fortunately, he wasn't working on development. So we, for a number of years, worked on this central question in T-cell development of how double-positive immature thymocytes give rise to either CD4 helper cells that are class II specific or CD8-positive cytotoxic cells that are class I specific. And incidentally, this remains a puzzle after almost 30 years. There are hypotheses out there, but certainly no definitive mechanism as to how the decision is made. We know about transcription factors that are involved, and it is through one of the transcription factors expressed in the thymus, ROR gamma T, that we got into what I'm going to tell you about today, because we found this molecule to be required for survival of the double-positive thymocytes. And in the study that Zuoming Sun did in our laboratory quite some time ago now, he found that animals lacking ROR gamma T not only had a defect in thymic development, but were also completely lacking lymph nodes, as shown over here, and they lacked a kind of cell that at that time we and Jason's sister called the lymphoid tissue inducer cell, but it was a cell that had been identified by Rena Mabius and Irv Weissman a few years earlier. You can see it here in a fetal mouse and staining with antibody against ROR gamma T in red and CD4 in green. And these are not T lymphocytes. These are innate lymphoid cells that weren't called that at the time, but they are absolutely required to induce the genesis of lymph nodes and Peyer's patches. And then later we found also for induction of so-called isolated lymphoid follicles through these structures called cryptopatches that are found in the intestine, particularly in the small intestine. This is just showing the villi in the small intestine with neurons running to the tips of the villi. So these structures here were what got us really interested in studying aspects of peripheral T-cell functions, and I'll explain to you how that happened. It was through work of Ivo Ivanov, a very talented and creative postdoc in the lab about a decade ago, because what Ivo did was he wanted to study these particular cryptopatch cells, and Gerard de Berel in the lab had made a knock-in mouse inserting GFP into the ROR gamma T locus, and that defined these lymphoid tissue inducer cells. These are tertiary lymphoid tissues that occur postnatally, and these are structures that are surrounded by dendritic cells that basically sense what's in the lumen of the intestine. And when these cells become activated somehow, they make a chemokine to attract B lymphocytes and form isolated lymphoid follicles. So Ivo was studying these cells and noted something that we had missed when we first analyzed these GFP knock-in mice, and that was that there were some GFP intermediate intensity cells shown here that express the T-cell receptor, the alpha-beta T-cell receptors. And it turned out, obviously, these were T lymphocytes, and he began to look at what the property of these T cells might be. These are the innate lymphoid cells, LTIs, down here, that are bright for GFP. And around the same time, Dan Kua, at what was then DNEX in Palo Alto, called me because they had been looking at cells that I had never heard about that are responsive to a cytokine called interleukin-23, and that make another cytokine called IL-17. And there were very few papers on IL-17 at that time. But he told us that in doing microarrays of those cells, he found upregulation of ROR gamma T. So we immediately looked in these mice, and we could see that in the GFP negative T cells, there's no intracellular IL-17, but in the GFP positive cells, you see most of them make IL-17, and if the animal is rendered homozygous null, and that is with GFP in both alleles, there's almost a complete loss of the Th17 cells of interleukin-17. So what are these cells? Well, we've known since the mid-1980s through classic work from Tim Mossman and Bob Kaufman, who are also at DNEX, that helper T cells, CD4 positive cells, can come in two different categories based on the kind of cytokine they produce. They can be interferon gamma producers. Those are Th1 cells that are required to kill a variety of different intracellular pathogens, but have also been implicated in autoimmune diseases. And there are Th2 cells that make the cytokines IL-4, IL-5, and IL-13. And these are required to fight off parasitic worms or helminths, but they've also been shown to be important in allergies and in asthma. And these cells themselves are polarized from uncommitted CD4 positive T cells through various cytokines, IL-12, which induces the transcription factor T-bet on these cells, in these cells, and IL-4 that comes from an innate source to induce through GATA3 the differentiation of these cells. So it was really only in the mid-2000s that these Th17 cells became apparent, and they're called Th17 cells because it was IL-17A and F that they produce. These are cells that are required to kill various extracellular pathogens at mucosal surfaces, both bacteria and fungi. They are critical for tissue repair at mucosal barriers, but on the other side, they also cause autoimmune inflammatory diseases. And initially, that was shown by Dan Kua in a model for multiple sclerosis in mice, but other models showed the importance of these cells, and then it became clear that these cells are critical in human autoimmune disease. And these cells differentiate in vitro through a variety of different stimuli. One can just stimulate with IL-6 and TGF-beta, and the TCR activation, the presence of those cytokines, polarizes to these cells, or another combination of IL-6, IL-1 and IL-23 leads to polarization of these cells into what might be called more pathogenic types of Th17 cells, and I'll come back to tell you about that. This, at the time of the discovery of the role of TGF-beta here, was very puzzling, because it was known that there's another type of CD4 positive T cell, and that is the regulatory T cell. These are cells that are absolutely essential for preventing massive inflammation. There are people who have mutations in the transcription factor required for Treg, FOXP3, and they suffer from fatal autoimmune diseases within the first couple of years of life. These cells depend also on TGF-beta for their differentiation or maintenance. And in particular, in the periphery, they can originate in the thymus, but they can also originate in the periphery as induced regulatory T cells. And I'll tell you a little bit about those as well. So in summary then, it turned out that ROR gamma T directs differentiation of multiple lineages. This is a nuclear receptor that I'm not going to tell you much about today, but it's required for the development of these lymphoid tissue-like, inducer-like cells in the postnatally. And these are cells that make cytokines, but they also lead to formation of these cryptopatches. And then gamma delta T cells that have gamma delta T cell receptor can also make IL-17 and are probably very important in some diseases like psoriasis. I told you about the Th17 cells. And then there are the type 3 innate lymphoid cells, which are related and can also sometimes are confused with the lymphoid tissue inducer cells. And these they can have inflammatory functions, but they are primarily important for maintaining barrier integrity. And I told you about these developmental processes in the thymus and in lymphoid organ development. So at the various barriers, the Th17 cells are critical for dealing with these kinds of microbes, the various kinds of pathogens shown over here. But as I told you, they participate in autoimmune disease. And in humans now, it is clear that these cells have roles in inflammatory bowel disease, and interleukin-23 is particularly important there. In various arthritides, the Th17 cells are important in ankylosing spondylitis, as well as we think in rheumatoid arthritis. And in psoriasis, there's no question that Th17 cells and gamma delta cells that make IL-17 are critical. And in fact, there are highly effective antibodies that are now in the clinic targeting interleukin-17 that are really tremendously effective. So I'll tell you a little bit about the Th17 cell, their differentiation, and at the end I'll tell you about a possible involvement of these cells in autism, and particularly in an animal model of autism, and I'll try to put these in the context of how the microbiota may function. Okay, so what led us into studying the microbiota was that Ivo, after he discovered the Rho gamma T Th17 connection, decided to treat animals with antibiotics. And he had found that Th17 cells are the most abundant in the small intestine in our colony, in the lamina propria. And he noted that there was a marked reduction in the proportion of Th17 cells in the antibiotic-treated mice. And then he looked at germ-free mice that we just kept in isolators. And you see they have essentially no IL-17 being made by their CD4 positive T cells. And then he made well, I should say, we've all heard about the microbiota. It's gotten a tremendous amount of press in the last few years. And we hear about more and more functions that the microbiota might have. There's some really beautiful papers out there, such as Jeff Gordon, who's really the pioneer in this field, who has shown the effects of malnutrition on the microbiota, leading to lasting changes in whole body metabolism. But also he's shown that discordant human monozygotic twins have microbiota that, when transmitted to mice, can basically phenocopy the obesity versus leanness phenotype. Sarkis Mazmanian published a paper with Paul Patterson on an autism model in mice, suggesting that microbiota in mice can contribute to neurological changes. And I'll tell you a bit about a different angle on this at the end of my talk. And then there's also been demonstration of the role of microbiota in the circadian clock regulation. But the way we really were able to start getting a handle on this was a very fortuitous finding that Ivo made. And he noted that depending on where he purchased mice of the same strain, he saw either very few Th17 cells in the intestine or a lot of Th17 cells. And we were very fortunate that we could collaborate with Kenya Honda in Japan, who had colonies monoassociated with various bacteria. And through that, we were able to show that the mice from Jackson from Taconic farms have segmented filamentous bacteria colonizing them. These are bacteria that are gram-positive anaerobes. They form spores, and they're not really related in terms of their sequence to any other bacteria that we know about, except very, very distantly to some of the Clostridia. And they're very, very difficult to culture. Sansonetti's group in Pasteur has been able to establish a system on growing these on epithelial cells, but they're very, very difficult to actually do any biology with and do any genetics with. But you can see that these bacteria form these segmented filaments. They embed themselves in the epithelium in the small intestine, and there they cause an actin polymerization reaction, presumably through secretion of effector molecules, but we understand very little mechanistically as to what these cells may do. But what we did see was that whereas in germ-free mice you don't see any CD4 T cells making IL-22 and IL-17, after about a week of colonization only with SFB, we see this massive induction of these Th17 cells, and we showed that this actually protects the animals from infection with pathogenic enteropathic bacteria like adhesive effacing E. coli and Citrobacter rodentium, which is a mouse pathogen. And much of the protection comes from the production of this IL-22, both by these cells, but now we also know by the type 3 innate lymphoid cells. But the flip side of this is that these cells, as I told you, can be pathogenic. And in collaboration with Diane Mathis and Christophe Benoit across the river here, we were able to show with Joyce Wu in their laboratory and Evo working together that in a mouse model of spontaneous arthritis that they had developed, the so-called KB/N mice, these animals do not get sick in a germ-free condition, but after colonization, just with segmented filamentous bacteria, the animals develop severe arthritis within about a week. So going from being normal like this to having these kinds of joints. So obviously, we also wanted to know whether something similar might be going on in human. But let me just show you that this is not the only example of an autoimmune model in which microbiota are important. This is another example that Takuya Uehara in our lab has worked on. It's a model for experimental autoimmune encephalomyelitis in which mice are injected with a myelin protein and an adjuvant, and typically within 2 weeks they develop various degrees of paralysis. And what Takuya found was that treatment of the mice with either ampicillin or vancomycin almost completely prevents the onset of the disease. And we are now basically trying to identify the bacteria that might be involved here, because these are mice from Jackson Laboratory and they do not have any SFB. We've tested for that. So it's, we think it's going to be a general phenomenon in which microbiota are important and we think are causal. Now, in human, Jose Scher, when he was a rheumatology fellow in our laboratory, studied several cohorts of patients at Bellevue Hospital, new onset rheumatoid arthritis patients who had not yet been treated, as well as people who had arthritis for years, who had had various different medications, healthy controls, and psoriatic arthritis. And what he found was that only in the untreated cohort, in their fecal microbiota, there was a preponderance of one type of gram-negative bacterium, Prevotella copri, and that this was found in roughly 70 to 75% of these patients, but in only 15 to 20% of healthy individuals or individuals studied in the Human Microbiome Project. And we also found that there may be some differences in sequence of the Prevotella from patients versus the Prevotella from healthy people. So we are pursuing this to try to hunt down whether there is some kind of a causal relationship here, but at this point we are still dealing only with correlation. Oops, I think I took this from another presentation here. So how do SFB-elicited Th17 cells in the intestine exert pathogenic functions systemically? So one of the things that we wanted to solve early on was the antigen specificity of these SFB-elicited Th17 cells, and then ask whether these cells can circulate to lymphoid tissues that are draining specific organs. And I'll just show you a little evidence or show you a bit of how we achieve this, looking at the specificity, but I'll tell you that we do find the induced cells to distribute broadly within the body. So Benny Yang in our laboratory took on the project to look at the specificity of the induced Th17 cells by taking T cell receptors, cloning them from single cells, putting them into a reporter cell that would turn green now when it recognized antigen presented by antigen-presenting cells from black 6 mice. And he was able to show that within the colonic content, the cells that he isolated, the T cell receptors he isolated, responded exclusively to SFB. And he used that to make MHC tetramers, as well as T cell receptor transgenic mice that express these particular TCRs, and was able to do experiments such as this, which is to introduce into mice naive T cells from these mice bearing a SFB-specific TCR, and then either feed the animals with SFB or not, and look in the lamina propria as well as at various inductive sites. And you can see here that if the animals are SFB positive, the donor-derived cells now expand, and they actually turn on ROR gamma T in the small intestine, whereas we don't see any expansion of these cells in SFB negative animals. So I'm not going to go through all the work that's been done since on this, but just to summarize what we now know. So SFB in the small intestine induces in the draining mesenteric lymph node the polarization of CD4 cells to express ROR gamma T. Then these cells can distribute widely and I'll show you a bit what they do in a moment. On the other hand, inducers of Th1 cells like Listeria monocytogenes, and we used an attenuated form of Listeria that's not adhesive, that can colonize the intestine. It still induced T helper 1 cells, and if we embedded in the Listerial genome a gene from SFB, for which we had the T cell receptor specificity that I showed you before, we got SFB-specific T cells that became Th1, showing that it's the property of the bacterium that determines whether cells polarize to Th1 or Th17. So that's one of the major questions in the field, is to understand what it is. Is it a niche in the microenvironment that favors the localization of particular types of antigen-presenting cells that can polarize then naive T cells here in the draining organ? Or are there inductive signals from these bacteria that basically alter the program of the antigen-presenting cells that then act distally here? And we've also studied regulatory T cells. Kenya Honda had shown that a group of Clostridia in mice and Clostridia from human can polarize colonic CD4 T cells to express FOXP3 to become regulatory T cells. And I'll show you in a different model with Helicobacter hepaticus that something very similar can occur as to what I showed you with SFB can occur with regulatory T cells. And then the question is, how is it that these cells that are polarized here participate in autoimmunity distally? So Mo Shu has done something very similar to what I described for SFB with the response to Helicobacter. I told you SFB colonizes the small intestine. It leads to Th17 cell polarization there under homeostatic conditions, but it does not promote colitis. It's a non-pathogenic bacterium, and it actually protects the host from pathogenic enteric bacteria. Helicobacter hepaticus, on the other hand, is very similar to human Helicobacter, which inhabits the H. pylori, which inhabits the stomach. And this typically does not cause any disease, but in certain animals, for example, those lacking interleukin-10, it can induce a pathogenic type of Th17 response, and it can induce a severe colitis, was shown by Fiona Powry. We wanted to ask, what is the T cell response under homeostatic conditions? So what Mo did was to identify T cells, Th17 cells, that were induced by H. hepaticus in mice lacking interleukin-10, made T cell receptor transgenic and other reagents specific for Helicobacter. And very similarly to what he did with SFB, he found that these cells reacted with H. hepaticus antigen. So what he did was to colonize mice with both SFB and H. hepaticus at the same time, and then introduced both the SFB-specific and Helicobacter-specific T cells that he could distinguish from each other in the recipient mouse. And as I showed you before, as I told you, in these animals, most cells in the small intestine that are specific for SFB become ROR gamma T positive cells that makes them Th17 cells. But the Helicobacter-specific T cells in the large intestine, you can see express ROR gamma T for the most part, but also express FOXP3. It turns out this is a property of induced regulatory T cells. They express FOXP3, but most of them also express ROR gamma T that contributes to a unique program that they have, and that was shown by Diane Mathis and Christophe Benoit last year, that they do have a unique gene expression program, distinct from other kinds of regulatory T cells. Those T cells that specific for Helicobacter that don't express these markers are follicular helper cells that help B lymphocytes, and these are particularly prevalent in the cecum where there's a lot of follicle formation. So what happens in an inflammatory condition in IL-10 knockout mice? So you see what happens here. These very same cells in the IL-10 knockout mouse now express ROR gamma T. Very few express FOXP3 anymore. And these cells can now participate in promoting colitis. and they're pathogenic Th17 cells. So what makes them pathogenic? Well, they're different in gene expression profile from these cells here. One of the features is shown here very clearly. And looking in the same mice with both types of cells in an IL-10 replete or IL-10 deficient animal, you can see that the Helicobacter-specific T cells upregulate not only IL-17, but also also interferon gamma, as well as the interferon gamma driving transcription factor T-BET. The 7B8 cells, the homeostatic, non-pathogenic T cells specific for SFB, do not, even though these animals are deficient for interleukin 10. So we're trying very hard to understand what's the distinction between these different programs of Th17 cells. So this summarizes what I told you, that there can be these homeostatic cells induced by SFB and these regulatory cells induced by Helicobacter that can become pathogenic under certain circumstances. So we've taken another approach as well, and that's based on some of the information that's been coming into the field from a number of people related to the role of the interleukin 23 receptor that's expressed on the T helper 17 cells. So it turns out IL-23 is required for these cells to become pathogenic and to turn on interferon gamma. And Gita Stockinger in London has done fate mapping experiments and has shown that cells that have gone through interleukin 17 expression can turn on, in the presence of IL-23, subsequently in vivo, can turn on interferon gamma as well as GM-CSF that participate in pathogenesis in autoimmune disease. But that requires IL-23 receptor. And in humans, something very similar seems to occur. In the circulation of CD4 positive T cells, Federica Salusto showed that there are cells that express the chemokine receptor for Th1 cells, CXCR3, as well as the chemokine receptor for Th17 cells, CCR6, and these cells make interferon gamma. But they're not classical interferon gamma producing Th1 cells. She called them Th1 star because they require ROR gamma T. And Jean-Laurent Casanova found several families in which there were mutations in the ROR gamma gene and showed that these cells are defective or do not develop, do not differentiate in these patients. And in fact, they are quite susceptible to infection with mycobacteria. Immunization with BCG can lead to disseminated mycobacterial disease. So these cells seem to have evolved in a way to deal with particular kinds of pathogens like the various mycobacteria. So Lin Wu has decided to look at IL-23 receptor as a distinguishing fact, to distinguish between pathogenic and non-pathogenic T cells. And we collaborated with Rahul Satija, who used to be here, who is now at NYU and at the New York Genome Center, to do single-cell RNA sequencing analyses of T lymphocytes in this case. So Lin has reconstituted mice with irradiated mice with bone marrow from IL-23 receptor heterozygous and IL-23 receptor mutant deficient cells. He then can distinguish between these different donors in mice in three different models: a pathogenic model of EAE, a pathogenic model of colitis, and the non-pathogenic SFB. And I'll just show you a couple of examples here. With the EAE model, looking at T cells from the central nervous system, CD4 positive T cells, Lin and Rahul were able to distinguish very clearly three populations of cells. And this population here is what we would call a homeostatic or regulatory population, amongst which we see FOXP3 and IL-10 being expressed. This population here is one that makes interferon gamma as well as IL-17A. It also makes GM-CSF with CSF2. And these are the pathogenic Th17 cells. And if he then looks to see the distribution of IL-23 expressing, IL-23 receptor expressing or non-expressing cells, you can see that those cells deficient for the receptor are excluded from this pathogenic population, but they're enriched in the non-pathogenic population. On the other hand, when he looks in the, and by the way, he sees something very similar in the colitis model in the gut. When he looks, though, at homeostatic conditions with SFB colonized mice, you see different kinds of populations. These are two populations of regulatory cells, including the induced Tregs here that make ROR gamma T. Then there's a more of a classical Th1 type population shown here that makes interferon gamma. And then there's this non-pathogenic Th17 population that makes interleukin 17A, but very few of these cells make interferon gamma, or they also make interleukin 23 receptor, as shown here. So we think then that there are these non-pathogenic Th17 cells that can differentiate in the draining lymph node, but under conditions where IL-23 signaling is prevalent, where IL-10 cannot inhibit this signaling pathway, these cells can become pathogenic. They can also presumably protect from various pathogens. And then there are these induced regulatory T cells which may have some kind of plasticity with various types of Th17 cells, but fate mapping studies on these cells have not really yet concluded where they actually come from. So what we currently think is that, based on a number of studies that I don't have time to tell you about, is that there are monocyte-derived cells that are required to detect the signal from SFB in the ileum. And within the first few days after colonization with SFB, these cells induce production of ROR gamma T in naive T cells. And these cells become Th17 cells that now can distribute broadly, going back to the ileum, but also in other places. We find them throughout the body, in the spleen and lymph nodes, distal to the intestine. And Teruyuki Sano decided to have a look at these, at how this induction occurs. And I told you that SFB is largely localized in the ileum. That's the only place it attaches. And that is basically shown here as we look along the length of the gut from duodenum through to colon. You can see on this log scale that it's in the terminal ileum where we see the most SFB. And we don't see any, of course, from these mice that do not have any SFB. And we had done a number of studies looking by RNA sequencing at epithelial cell responses. And we found that only in the ileum we saw upregulation of a number of genes, of which the most prominent were the serum amyloid A proteins, SAA1 and 2, which occur only after colonization with SFB. And then we also looked at the expression of ROR gamma T and interleukin 17, in this case using a reporter mouse with GFP reporting on IL-17A expression. And what was our surprise was that if we looked throughout the length of the gut, we saw very efficient recruitment of ROR gamma T positive cells in all regions. But only here in the terminal ileum, where we see colonization and SA induction, is there upregulation of interleukin 17. So we wanted to look at this a bit more closely, and I'll just give you the bottom line of what Teruyuki found. What he found is that upon colonization with SFB, there's essentially a short circuit within the lamina propria of IL-23 made by macrophages, by monocyte-derived cells, which acts on these type 3 innate lymphoid cells, the ROR gamma T dependent cells I told you about, and these cells then make IL-22. And IL-22 acts on its receptor in the epithelium, activating phosphorylation of STAT3, and then that's what's required for inducing transcription and then release of the serum amyloid A proteins. And this all happens within the first 3 days after SFB colonization. In the meantime, these antigen-presenting cells go to the draining lymph node, and there polarize naive T cells specific for SFB to express ROR gamma T. And these cells now, as I just showed you, can distribute quite widely throughout the length of the gut as well as elsewhere. But it seems to be here locally that you see IL-17 produced, and we showed that the serum amyloid A proteins contribute significantly to this induction of interleukin 17 within the first week of colonization. And these results led us to this model shown here that there's a two-step process to activate Th17 effector functions with this particular commensal bacterium. The first is a specification of the Th17 program, which we monitor by ROR gamma T expression. And this requires these antigen-presenting cells coming from the lamina propria. And these prime cells then need to be induced in the second step to be able to have effector functions. And serum amyloid A serves essentially as an endogenous adjuvant to provide this function. Now, we don't know how SAA influences the transcriptional program, but it allows us to make some hypotheses, one of which is that perhaps there may be weak self-antigen agonists that in the presence of these kinds of endogenous adjuvant function might be able to lower the threshold for activation of Th17 cells, thus leading to pathogenesis. So this and others are testable hypotheses here, but we have wanted to look at the role of serum amyloid A in particular, and these are apolipoproteins associated with high-density lipoprotein in the plasma. They're produced mostly in the liver during the acute phase response, for example, after infections, and there can be enormous increases in the serum. They're very highly conserved through evolution. There are a number of receptors that have been identified, putative receptors. To my mind, it's still unclear exactly how the SAAs worked. TLR2 and CD36 and a formal peptide G protein coupled receptor have all been identified as such. And they also can form complexes with retinol, which act in its transport. So we wanted to see how SAA may act. And previously, we had shown that SAA can function through myeloid cells. But it turned out we probably were using not very effective forms of recombinant SAA protein. And when we got our hands on good recombinant protein, we were able to get results such as these, showing that in the presence of just IL-6, which induces very little IL-17 by itself, SAA1 in the medium now can promote the induction of IL-17. And under non-pathogenic and so-called pathogenic Th17 differentiation conditions, you can see even without any difference in ROR gamma T expression, you see tremendous increases in IL-17 induction in the presence of serum amyloid A. Basically, this is consistent with the idea that there can be a priming and then a second step that is acted on by the SAAs. So June Lee, who did this work, wanted to see whether the SAAs have a pathogenic role in an inflammatory disease. So he used the EAE model that I already described to you, which is known to be a Th17 dependent disease model. And he also used CRISPR to generate triple knockout mice that have also a mutation in SAA3 that I'll tell you briefly about. And what he sees is that there is a marked attenuation of disease score in the SAA triple knockouts. There's also an attenuation in double knockouts for SAA1 and 2 or for SAA3 singly. And he wanted to see why that might be. And he looked in the serum, and what he saw was that after the induction of the EAE very quickly, there's elevation of SAA1 and 2 levels. And indeed, it's only in the liver here that we see induction of SAA1 and 2, but SAA3 is not made in liver at all. On the other hand, if we look in the central nervous system where the disease manifests itself, we don't see any SAA1 and 2, but we see elevation of SAA3. So we think that these cytokine or these adjuvant-like molecules contribute in different places in this particular disease models. And then June was also able to track these mice through the chronic phase of disease after 3 weeks. And he saw that some animals recovered pretty well while others did not, and looked at their serum SAA1 and 2 levels, and he saw a very good correlation here between disease score and the level of serum SAA, really suggesting very strongly that the SAAs do contribute to the ongoing disease. So this then brings us back again to this two-step model of effector T-cell mediated pathogenesis, in which we think that the microbiota can contribute early on for the induction of Th17 prime cells, but then this second signal, this adjuvant effect, is needed to generate effector cells, and we don't understand still what generates homeostatic versus pathogenic types of effector cells. And then these cells can participate in autoimmune disease. We think they can participate in various responses to tumors, and I'll show you the last thing that I'll tell you about this model for MIA, maternal immune activation mediated neuropathology. But we now have a lot of evidence emerging that the composition of the microbiota can contribute to how cancer patients respond to so-called checkpoint blockade immunotherapy, basically blocking what have been called the brakes on the immune system, and thus restoring responses against tumors. So there's a lot of excitement in this area. And we think that the microbiota have influences on autoimmune diseases that may resemble the influences that they may have on tumor-specific immune responses. But this is still a big question mark here. We're trying to understand mechanistically what different components of the microbiota might do. So very briefly, in just a couple of minutes, let me tell you about this model of maternal inflammation that can contribute in an autism model in mice. Now, the reason I mention this is because this is microbiota dependent, and as I'll show you, it's Th17 dependent. Going back a few decades, there have been suggestions that mothers who have had various types of infections during pregnancy in the first or second trimesters have a higher incidence of children with autistic behavior. This is still somewhat controversial, but people have developed some models in animals, and this is a model in which either viral infection or activation of toll-like receptors with poly-IC leads to maternal inflammation when this occurs during mid-gestation at day 12 and a half in embryonic life. And that leads to a very, what we see as very robust behavioral changes and developmental changes in the offspring. The champion of this model over the last couple of decades has been Paul Patterson, who was at Caltech and who passed away a couple of years ago. But I was alerted to a paper that came out a decade ago from Paul's lab that interleukin-6 in the mother is required for this particular phenotype. And I was very fortunate that when we found out about this, it was right after we had found the connection of ROR gamma T to Th17 cells, and Gita Stockinger had shown the role of IL-6 in differentiation of Th17 cells. So we proposed, Jun Hu in the laboratory as a side project, got interested in working on this, and what really made this possible was that his wife was a postdoc with Richard Axel at Columbia and studying neuroscience, and she got interested too, and she would come in and help us with behavioral assays at the outset, but then actually had a much more important role, as I'll show you. And we got funding from the Simons Foundation for Autism Research to take a look at this, to test the hypotheses that Th17 cells might be involved here downstream of interleukin-6. Now, there are three core symptoms of autism spectrum disorder: social interaction deficits, impaired communication, and repetitive behaviors, and all of these can be modeled in mice. After injection of poly-IC, you can see there is a rapid induction of interleukin-6. I actually don't have the time course. I do. Yeah, this is very rapid here, but then it drops off by 24 hours. But IL-17 goes up, IL-17A, and that is sustained beyond 24 hours or so. You can see here that in mice lacking ROR gamma T, specifically in their alpha beta T cells, there's no upregulation of interleukin-17. What we also noticed was that the receptor for interleukin-17A is present in the fetal brain. And you can see that in animals that have not been exposed to maternal inflammation, you can see some low-level expression in some cells here, but it's actually upregulated after poly-IC treatment of the mother. And if we now look at the offspring, in this case for repetitive behavior, which is bearing of marbles, you can see that animals treated with poly-IC wild-type mice have a tremendous increase in the repetitive behavior patterns, but animals that lack ROR gamma T, if the mother lacked ROR gamma T just in her T cells, we basically prevent this phenotype. Very similarly with social behavior, animals interacting with another mouse here in this chamber or with an inanimate object over here. Typically, the animals will spend twice as much time with another mouse than with an inanimate object. And what you see here is that after poly-IC, there's this reduction in interaction with the social animal, with the social target. And this is reversed either by anti-IL-17 in the mother or by the lack of ROR gamma T in her T cells. And that's shown in a heat map over here. And what was really quite remarkable was that June and Gloria Choi, his wife, in their new laboratories, have found that there is a very stereotypical kind of developmental defect in which these animals, the fetus here just, I forget what day this is, 18 and a half, I believe. Oh yeah, it's shown there. You can see that there's this defect here in the development of the cortex when stained with antibodies against proteins expressed in different layers. And this is prevented by anti-IL-17 in the mother. And they're also able to show that injection of IL-17A transuterine into the fetal ventricles basically can lead to behavioral phenotypes like I showed you. In this case, I show you a communication defect of ultrasonic vocalization. So this phenotype was seen only with IL-17A induction, not with IL-6 injection, and not if the fetus was deficient for interleukin-17 receptor. And similarly, you can see that there was the induction of this kind of a patch phenotype in the central nervous system, in the cortex. And so what we think, that's all the data I'm going to show you, but what we think then is that there is in the mother a predisposing condition here that we know requires induction of Th17 cells in the gut. I didn't show you the data, but that's where the microbiota are critical. And then in this case, signal 2 is a very artificial signal, effectively what would be something like a viral infection, and that leads to IL-17 induction. Somehow IL-17 gets across the placental barrier, leading to the behavioral abnormality and the cortical phenotype that I showed you, and that's dependent on the IL-17 receptor. So that raises a number of very interesting questions. What is the IL-17 receptor doing normally in the brain? Is it also expressed in similar types of cells in the human brain? And finally, do maternal infections cause activation of Th17 cells in a subset of susceptible women? And there could be women who have a particular microbiota similar to the mice that have SFB that predisposes them to have a high level of Th17 and production of IL-17. And we're currently looking at people who have, at the microbiota from people who have highly divergent levels of circulating IL-17 to see if there may be differences in the microbiota there. So I showed you that there can be both good and bad sides to the microbiota. We're undoubtedly going to learn a lot more mechanistically during the next few years, and it's definitely an exciting field where the kinds of tools that we now have, both for microbiology and for the immune system, are going to come in handy. So these are the heroes of the Autism Project, June and Gloria Choi. Gloria is at the McGovern Institute at MIT now. June is at UMass. And Chuck Hofer, who is now in Denver but was at NYU, helped us at the beginning with behavioral assays. And these are the folks in the lab who did the work that I described today, and some of those who did the work that led up to it. Raul Satija, our collaborator on single-cell analysis, Frederick De Beer provided us with the SAA double knockout mice, and Kenya Honda and Koji Adarashi helped us on multiple projects that we've collaborated on, and Mark Jenkins helped us with tetramers that I didn't show you. So thank you very much for your patience, and congratulations, Jack, on having a truly remarkable and inspiring career. I am just so honored to be here, and I thank you all. I'm happy to take questions.
Questions?
If you treat the mice with either very specific antibiotics or broad spectrum antibiotics, what would you find?
What do you see in terms of the autism model?
Yeah.
Well, this is no longer our work, but I can tell you that June is continuing this in his own lab. I can tell you that the specificity of the microbiota is critical. Okay? So just like I showed you with the EAE model, you can completely prevent the autism phenotype if you use the right conditions, the right antibiotics. And this also explains why this model, we didn't have an easy time getting this paper published. And that's because it was reviewed by neuroscientists. And some of my best friends are neuroscientists. I hope some of them are in the audience here. But neuroscientists typically do not care very much about the microbial health status of the mice in the colony. And that's absolutely critical. And nobody had seen such robust, reproducible results in this kind of work. It turns out if you keep your colony with the right kind of microbiota, you can get enormous reproducibility. That had just not been observed before.
Tim?
What kinds of co-receptors do GH17 cells use? What kind of T-cell receptors do they have? And how do the filament segmented bacteria, get to that, present the antigens to them?
All good questions. We don't know how the bacteria how the antigen is processed. Is it coming from dying cells that somehow engulf the bacteria and get taken up? We believe that CX3CR1 positive monocyte-derived cells are very important there. CD103 positive dendritic cells are not essential. We can completely get rid of them. But there could be some dendritic cell population that is important, not only the monocyte-derived population, for the presentation of SFB antigen on MHC class II. In terms of co-receptors, I'm not exactly sure what you're referring to. These are all CD4 positive T cells, but there can be CD8 cells, although we haven't seen those to SFB. CD8 cells have been largely neglected in the microbiota, in the gut in response to microbiota, but now people are becoming interested in them. Yeah.
So the link between SAA and CD36 is interesting.
Yeah.
CD36 knockouts, do they become more
No, CD36, well, we only looked in that polarization assay that I showed, and we used CD36 knockout T cells, and there's no difference in induction of IL-17. Same with toll-like receptor 2, same with the FK receptor, the formal receptor 2. FPLR2, I forgot now. So we don't really know. We think there may be some connection to transport of cholesterol or sterols, which is interesting in light of some work of Vijay Kuchru's, suggesting that lipid biosynthesis may distinguish between pathogenic and non-pathogenic Th17 cells, but it's still early days, so it's hard to tell.
One more question.
Susan.
I'm not in immunology, so I hope this isn't a clueless question, but if I think about some of these responses, there's a whole age dependence. So I was curious if there's a change as an organism ages with IL-17, or is it the exposure to different kinds of bacteria over time, or do you know anything about that?
Well, people have been doing studies on microbiota longitudinally, and there are changes with age in the microbiota, and that may contribute to changes in the immune system, which is known also to occur with aging. But again, it's very little that I've seen that, I could give you a really firm answer as to what's going on. I don't know if people have done this in mice. I have not seen that.
All right, well, I'd like to thank Dan for delivering the first Strominger Lecture, and congratulate Jack once again on the illustrious career, and that's why we're here. Thank you.